This week we will talk about a simple T-cell model. Once again, we're not discussing the immunology per se. We're only using an ideal simplified model. I must tell you that the T-cell is actually a very complex topic. This is not a course in immunology, so I will not go into details. But the point I'm trying to make is how we can model a process of inflammation. And inflammation can be caused by many different players. But we're supposing some kind of ideal T cell and describe what goes on in this ideal system that causes inflammation. Immune cells, also called lymphocyte, and I think you are already familiar with the B cell and T cell concept. And among T cell, we have two major different kinds, which is T helper cells and T killer cells. These can be sometimes called CD8 plus, CD4 plus cells, depending on the expression of what you call the cluster of differentiation, or CD. So immunologists talk about a lot of CD something something, but it's really just a jungle of CDs. We have so many different kinds of T cells. One grossly simplified idea how to understand B cell and T cell dynamics is that when there's an antigen, some foreign molecule, it should be identified and tagged such that the macrophage can come over and simply eat them and remove them. B cell and T cell interaction is basically about this tagging process. Sometimes T cell can even kill the cells by injecting, figuratively speaking, some kind of toxin. But that's called the T killer cell. But more interesting dynamics can be found in T helper cells too. Immunology, because of this complexity, has used a mathematical modeling to understand and simplify these processes. And one thing we are going to consider in this model is the process of detection of some inflammatory molecule that triggers the intracellular processing, signaling, and leading up to the gene expression that express certain inflammatory molecules such as the prostaglandin, and the whole process can be simplified in this way. This is a grossly simplified T-cell model. An LPS, lipopolysaccharide, is an inflammatory molecule, and it binds to the toll-like receptor, TLR, and when it's bound, it triggers this pathway. NF-kappa B is a transcription factor that can enable transcription of certain protein that can produce prostaglandin. Prostaglandins are the ones that cause various inflammatory reactions, such as the fever and drowsiness. Interestingly, in the early history of sleep research, prostaglandin D2, one kind of prostaglandin, has been proposed to be a somnogen that causes sleep, because the expression of prostaglandin D2 in particular can be robustly circadian. The answer is not clearly known, but it remains to be a very interesting possibility. In any case, LPS binding on TLR can dissociate the inhibitor of NF-kappa B, and NF-kappa B complex to dissociate. When they dissociate, the inhibitor of NF-kappa B will degrade. But the NF-kappa B can be activated, can come into the nucleus in a process called translocation. And bind to the promoter upstream to a gene such as COX-2 to produce prostaglandin. And we know that the limiting process for prostaglandin is the gene expression process. It is not intermediate process for accumulation and release of prostaglandin, but in fact, the gene expression itself is a key timing process for the release of prostaglandin. So all of these steps can be described by the compartment model that we have already used for epidemiology.